Welcome to the CIBM Center for Biomedical Imaging Breakfast Science seminar. Today's session will be chaired by Dr. Elda Fiske-Gomez, who is a research staff scientist at the CIBM Signal Processing CHUV EPFL, Computational Medical Imaging and Machine Learning section.
Hi everyone, good morning. So I will be introducing our speaker on the breakfast science today, who's Erick J. Canales-Rodríguez. So I will start with what he was putting on his on the ad of the presentation. So Erick's got a bachelor's degree in physics from University of Lausanne in Cuba, and a master's degree on the from the Faculty of Medicine at the University of Barcelona in Spain, and a PhD of the University of the Basque Country in Spain. He moved around. Didn't know that. He has published over, if you see, I checked on Google this morning, and then he has more than 80 papers. And if you add on the conference abstracts and everything, he sum up over 165. So he's an outstanding lecturer, I may say. I'm one of the humblest person I know, because with these records, it's kind of surprising. He has, okay. His research lies in the interaction between physics, signal processing, and computer science and neuroscience. He's been very active in the field of diffusion MRI imaging. When I first met him, I knew him as the diffusion guy, and he's been he has contributed mainly on signal modeling, signal reconstruction, and I can say some papers here. He gave a mathematical description of K space, he a Bayesian workflow framework to identify intravoxel diffusion, multi-tensor fitted-gated LDF estimations. He worked a lot in the convolution of the diffusion spectrum signal, and his interest slowly shifted down from diffusion to other contrasts to detect microstructure. So he's now an Ambizione Fellow. Is hosted at the Signal Processing Laboratory 5, and he works mainly on myelin mapping. So his abstract is like the axon myelin unit is essential from the normal function of the brain, that we know everything. So everyone. So now I think he will be presenting more on myelin imaging, axonal myelin mapping. So I think I let you introduce yours, Erick. Thank you very much.
Thank you very much for the introduction and invitation. It's a great pleasure to be here to show you our results. So I will talk about myelin and axon radius, which are two of the most important microstructural parameters of the white matter. Both determine the speed of action potentials and are involved in several ranges of science. We have been developing and working on different types of techniques to estimate these two parameters. I will split the talk into two parts. So in the second part, I will introduce a new surface-based relaxation model to estimate axon radius in vivo. And in the first part, I will show you some of the algorithms that we have been developing to estimate the Mallet-Water fraction using multi-component T2 relaxometry. So basically, the T2 relaxation time is the time constant, quantifying how fast the transverse manifestation vector decays because of spin dephasing owing to local field disturbance and dipolar interaction. In brain tissue, and in particular in the white matter, there are different water pools with different T2 times. For example, the T2 time of free water or cerebrospinal fluid is around 2 seconds, while the T2 of the water in the intra and extra-axonal compartments is more shorter, is around 60-90 milliseconds, while the T2 of the myelin water filling the space in between the myelin layers surrounding the axon is more shorter and is in the range from 10 to 30 milliseconds. And the goal of multi-component T2 relaxometry is to try to compute the volume fractions and the mean T2 values of all these pools and any other pool that we can have in brain tissue. So basically, to do that, we need to make some assumptions, not too many. So the first one, for example, is to assume that there is an underlying distribution of T2 times in the voxel that give the fraction of microcompartments with a given T2. And then we also assume that the signal that we measure in the scanner is the sum of signal from all these microcompartments. So basically, then we have this mathematical model where we model the signal for different echo times as the integral of this distribution of T2 times multiplied by the exponential relaxation model, that is a classical relaxation model. And as we are not assuming any parametric form, so we are not assuming any Gaussian or gamma distribution, for example, the goal is to estimate the no parametric shape of this distribution. And to do that, we discretize the integral. Basically, we define a grid of T2 times in the range typically from 10 to milliseconds to 2000 milliseconds. That is the range of T2 times in tissue. And then the goal is to estimate these weights that are the amplitude of the distribution evaluated at the discrete point on the grid. In general, solving this problem is challenging because in general we use many more points in the grid than the number of data points that we measure in the scanner. Then when we estimate the distribution in a given voxel, for example, this is a typical shape that we obtain. In general, we have a distribution with different lobes that in general don't overlap each other. And this lobe can be classified according to the range of T2 time. For example, the dominant lobe with a T2 time in the range from 40 to 200 milliseconds is assumed to be the intra and extracellular water, and the lobe with the shortest T2 time in the range from 10 to 40 milliseconds is the myelin water. Then from this spectrum, after this classification, we can estimate the myelin water fraction, that is just the proportion of water molecules in the myelin layers in relation to the total number of molecules, water molecules in the tissue. And to do that, basically we compute the area under the curve of this lobe and the total area of the whole spectrum. And this is, for example, a myelin water fraction map from a control volunteer. And this is the typical distribution or spectrum that we get in a given voxel, for example, here in the frontal lobe. In practice, for the multi-echo spin echo and multi-echo gradient and spin echoes that we use to reconstruct the model, the standard exponential relaxation model cannot explain the experimental data because this model is based on the assumption of ideal refocusing pulses. However, in the scanner, we have in general inhomogeneities in the big one field, and this creates some artifact or distortion in the signal. For example, here I am plotting the signal that we measure in two different voxels. In red, you can see that the signal decays following more or less an exponential or multi-exponential model, while in the frontal lobe, where in general we have higher inhomogeneities, the signal strongly oscillates, especially at the beginning of the decay. So basically, we cannot use this model because the model will not be able to fit the data. In this case, we need to replace the standard model based on the Bloch equation by the EPG model, is the extended phase graph. That is more general and is employed to describe relaxation processes with non-ideal refocusing processes. In this case, now, after having this more general version of the signal, we need to estimate both the distribution of T2 times and the actual refocusing flip angle in the experiment that is different from the flip angle that we prescribe it in the scanner. Then the next step is to discretize integral, and for that, the standard way of doing that is just by using a logarithmically equidistant spaced T2 values to have more points closer to the myelin, because this component decay very fast. And then basically with the EPG model, we create a dictionary of MRI signals, and then we collapse this beam width with the amplitude of the spectrum that we want to estimate. And basically what we estimate is this multiplication, that is the area under the curve of the spectrum for that for each particular beam. And then to solve the problem, we use non-negative least squares because we are estimating volume fractions, so we cannot have negative values. And however, in practice, this problem is very ill posed because the correlation of the columns of the matrix is very high. And so the problem is quite sensitive to noise. And to get more stable solution, we use regularized non-negative least square. And that basically is this equation. We complement the standard least square error by this regularization term that help us to introduce some type of a priori information. For example, by defining in a proper way this metric L here, and also by choosing the right regularization parameter that control a trade-off between the fitting, the term that describes the quality of the fitting, and the term that describes the a priori assumption that we can make about the solution. In general, we use metrics here to promote smooth solutions, because we don't want to be very invasive about the typical solution. Then this problem can be straightforwardly rewriting in this way, so that now we can solve the regularized NLS just by using the same solver that we used to solve the standard NLS. Then one of our first contribution in this topic was to review and compare different no-parametric algorithms to estimate the T2 spectrum and the myelin water fractions. And basically, we employed different regularization terms to promote smooth solutions and different methods or criteria for estimating the regularization parameter. Then, in practice, we used 3 different metrics to promote smooth solutions. The first one is the identity metric, and the other 2 are first order and second order Laplacian that are linked to derivatives. So basically, they penalize solutions that oscillate too much. So we promote a smooth solution that way. And then to estimate the optimal lambda, that is the regularization parameter, we employed the K residual fitting criteria that is very standard, is because it was invented by Alan McKay, the guy that invented this technique. And so for this reason, it's the more popular, but it's not necessarily the best, as you will see later. We also employed the L-curve. That is a very heuristic way to find the optimal regularization parameter. Basically, we plot here, so we estimate the solution for many, many different values of lambda. And then we plot the log of the residual versus the log of the regularization term. So the optimal lambda is just a point around here because it produces a good trade-off between both solutions. So for example, if you use a lambda equal to 0, the residual will get the minimum value, but the solution will not be close to the prior expectation that we are assuming. However, if we use a very strong lambda, then the solution will satisfy the condition that we are assuming for the distribution, but then the residual will be high, so we are not taking into account the data. So basically, a point around here is something that is in between both terms. And finally, we also considered the generalized cross-validation that is quite standard in inverse problem. Basically, the optimal lambda is the one that minimizes this term, that is the residual, and in the denominator, you have a term that penalizes the complexity of the model. So it's the idea. And at the end, we have 10 different methods because we have the combination of the 3 regularization matrices and the 3 criteria for estimating the regularization parameter, plus the standard NLS. Then we generated many different numerical simulations to compare all the algorithms to see which one was the best. And for that, we generated a mixture of Gaussians with different amplitudes, myelin water fractions, different position for the lobes of these Gaussian mixtures, different standard deviation, different flip angles that we may have in real data, and different SNRs. Moreover, we did a small rescan reproducibility analysis. So we scanned 5 subjects twice using the multi-echo gradient and spin echo sequence developed by Siemens, the Siemens group led by Toby. And after considering all the results from the numerical simulation and the reproducibility analysis, we have some more a list of recommendations if you want to use this type of technique in your studies. The first thing is that there is not single optimal method for all different noise level. So the optimal method depends on the noise level that you have in the data. So for the reason, the optimal method may depend on the scanner, how big is the noise in the scanner, or the acquisition sequence, or voxel sizes, acceleration factor. So many things that can affect. But if you, for the reason, we recommend to do a scan analysis to see what method is more reproducible in your for your data. And then if you cannot do that because you already acquired the data, but you are mostly interested in studying the milliwatt fraction, then you can choose the L curve and the K-square method using the identity metric, because they were top performance in the numerical simulations, and L curve was more reproducible in the scan analysis. On the other hand, if you are interested in computing the whole T2 distribution, not just the milliwatt fraction, because from the distribution we can compute other many different metrics, then we recommend to use the K-square method using the first or second order Laplacian metric. And all these methods are implemented in this toolbox. It's a Python toolbox that is very easy to use. So if you want to apply this, you can check our web. Then our second contribution in this topic was to revisit the T2 spectrum imaging inverse problem and to propose a new Bayesian regularized non-negative score method. Basically, we realized that there are 2 different ways to discretize integral, which are similar but not exactly equivalent. For example, here you can see that on the left side, I am using the standard way that I showed you previously, where basically we multiply the beam widths and the amplitudes of the distribution that we want to estimate. And basically what we are estimating is the multiplication of these 2 factors, that is the area under the curve. So basically this is the area of the beam. However, we can, as we already know these values, we can multiply these beam widths and the EPG model of signals. And so basically we have here an alternative solution in which what we are estimating now is the intensity of the spectrum, not the area. So these 2 solutions are actually equivalent. We can change variables and we can go from one problem to the other. It's equivalent. But the thing is that they only are equivalent when we don't apply any type of regularization. So if now we regularize, for example, this is the standard form. When we include this regularization, basically we're promoting solutions with a minimal area under the curve. However, if we here add this regularization using the identity metric, like here, it is possible to show that solving this problem is equivalent to solving a problem like this in the standard form, not the alternative form, but replacing the identity by this metric that is just codifying the distance of the beam widths. And as a result, basically what we are minimizing here is not the area under the curve, but the amplitude of the spectrum. So both solutions are more or less similar, but are not the same. And the thing is that any researcher reported previously the way in which they created the inverse problem. And this is important because then results are not reproducible. Even if the solution may be similar, they are not exactly the same. And this is an example just to show you that in fact the solutions are not the same. I created these ground truth spectrums, all are identical. Then I generated the signal for all of them using different noise levels and different noise realizations. And this is the solution resulting from the standard discretization form. And this is the solution resulting from the alternative form. They are not the same. So we decided to do a numerical simulation to compare these two methods. And also we proposed a new Bayesian method to automatically estimate the regularization parameter in the solution. I will not give you the details now, but you can email me if you are interested, or we can talk about this later. Then we compare again the different methods. We are now comparing the standard regularization form versus the alternative regularization form. And then we included the two best method in the previous, found in the previous paper, and the Bayesian method proposed in this paper. Like in the previous paper, we generated many numerical simulations to study the stability of the algorithms using many different distribution, noise levels, etc. And in addition, we conducted a validation study using histological data and in vivo MRI data from a dog spinal cord, from which we already know the myelin volume fraction. So it's important to know that what we are estimating is the myelin water fraction that is correlated to the myelin volume, but it's not the myelin volume. Because in the scanner, what we can measure is the water and how the water interact with the cell. So these are two different metrics, but they should be correlated, because if you have more myelin layers, you will have also more myelin water, right? And then this is one example of the linear relationship between the histological myelin volume fraction and the estimated myelin water fraction, estimated using the new method by Streck, using the alternative regularization form. These plots correspond to the white matter, gray matter, and white matter plus gray matter. The correlation is really good. These are actually the highest correlation reported today for this ex vivo MRI and histological dataset that I forgot to mention are free. So you can download the data in this on this web. And after taking into account, considering all the solution from numerical simulation, the validation using histological data, and also some real data that I am not showing today, basically we found that in general, the alternative regularization form, so basically the new one, is better in general than using the standard form, and also that Bayesian is a good alternative for estimating T2 distribution. It is not producing the best solution in all possible cases, but is the solution producing the best solution in a higher number of cases. So it's the more stable against different level of noise. Then, to conclude that part, I would like also to show you the work of Thomas Yu, a former and brilliant PhD student in our lab, supervised by Jean-Philippe, Mary, and Toby. Basically, he had the brilliant idea of using machine learning, of course, to improve the estimation of the inversion. And basically, what he did was he trained a multi-layer perceptron, and he generated thousands, well, millions of MRI signals using using the EPG model, and these signals are already generated from millions of different T2 spectrum with different number of lobes, different position, different standard deviation. And after doing the training, the estimation is really fast. Basically, the network takes as inputs the MRI signal that in general has 32 echo times, and output the associated T2 distribution in the logarithm space that usually has like 60 or 100 points. And this is a figure showing how stable it was the new method that is here, machine learning, modeling form machine learning. Basically, he generated a single distribution and many different signals from that distribution using different noise levels. So in theory, if the method is stable, you should get always the same distribution. And this is basically what we got here using the machine learning method, while with the standard conventional regularized non-negative least square methods, the method is in general stable in detecting the dominant lobe, but the myelin water flow lobe is very variable. So it's quite difficult to locate the right position for this lobe. And we did the same in real data. Now I'm showing the spectra from 2 different subjects, a single slide per subject. And you can see that in general, well, actually in general, when we get this type of result, we say, okay, there is a lot of variability in this lobe. Perhaps this is real. So perhaps the T2 of the myelin water is different, but perhaps that is not the case. Perhaps it's just that what we are noting is just the variability related to the reconstruction algorithms, not just that the T2 is so different, because with the machine learning approach, we estimate 1 or 2 lobes, but they are very close and consistent. And this is an example comparing the myelin water fraction maps estimated using the machine learning methods and the standard regularized NLS methods. As you can see, this method is able to detect the myelin water fraction in the frontal lobe, that in general is difficult. As you can see here, we cannot detect the right values in many voxels. And that happened because in the frontal lobe, the myelin water fraction is smaller, because the number of myelinated axons is smaller also. And it's also the region where we have more B1 inhomogeneities. And then if the flip angle is not estimated properly in the first step, then there are some oscillation in the signal that we cannot model, and this affects the final estimation. And then in the second part of the talk, I will present something very different. Now we are using diffusion relaxation model to try to estimate axon radius in vivo. Basically, the main motivation for that is that axon diameter is a fundamental parameter that till now only can be estimated in vivo using diffusion MRI data. However, now we know that there is a resolution limit below which we cannot estimate axon radius. For example, signals from axon smaller than 2 micrometer are very similar, the diffusion signals. And for the reason we cannot differentiate, we cannot distinguish one from other. And in practice, most axons in the brain have diameters below that limits. So this means that the current methods that are now estimating indices of axon diameter, what they are estimating is the diameters of the small population of axons with huge diameters. So basically, the method is not sensitive to smaller axons. So for the reason, it is desirable to identify additional MRI contrast source that can be sensitive to the smaller axons. And then, on the other hand, in the physics of porous media, it is well known that the interaction between the water molecules and the surface of the confining geometry reduces the T2 realization time. And in fact, this type of surface-based realization model is employed to estimate porous Rs in many materials. And so basically our idea was to say, okay, let's try to see if we can estimate axon diameter using these physical models employed in physical forest media. And then we designed an experiment to estimate the interactional T1 and T2 times. And we also used histological data to investigate whether these interactional realization time are related to the histological radius, and also to see if we can predict axon radius using this type of model. And this is a graphical illustration of the experiment that we are doing, adapted from Marco Pisolato, a great collaboration. Basically, when we used a diffusion MRI in the scanner, the idea is that the signal that we measure is affected, or is a combination of the signals coming from the interactional space and the extractional space. However, if we use a very strong diffusion weighting, that is a v-value in vivo data, a v-value higher than 5,000 seconds per millimeter square, basically we highly attenuate the signal coming from the extractional spins. And so basically we almost have information about the interactional axons. Then, but the thing is that this signal jets depend on the orientation and the distribution of the axons. But then if we take the spherical mean, that is the mean value of the diffusion MRI signal measured for all these gradient directions, we get a new value that is rotationally invariant. That is, the spherical mean coming from this sample is the same that the spherical mean coming from this sample where basically we reoriented all the axons, and now all the axons are parallel, right? And for that reason, the spherical mean, what it's providing is information about the typical axon, the average axon in the sample. Then if we repeat that experiment using different echo times, basically we can measure the relaxation time of the average axon in the sample. And basically this is what we did. We implemented a diffusion T2, T1 weighted MRI sequence, where we used diffusion MRI, and we used many different echo times and inversion times. We compute the spherical mean for all the 48 gradient direction that we used using a very high p-value. The data was acquired in the Connectome scanner at Kubrick in collaboration with Erick J. And then here you can see the spherical mean signal from the 9 volumes that were acquired. And now we use these 9 volumes and this diffusion relaxation biophysical model to estimate the interactional T1 and T2 relaxation times, and this linear constant that is proportional to the interactional volume, the proton density, and also is affected by the diffusivities, but we are not estimating this parameter. We don't want to disentangle this parameter now. We just are estimating the content because we are interested just in the relaxation times. And then we need to, of course, we need to relate the interactional relaxation times and the axonal radius. And for doing that, we are using this surface-based relaxation model that is adapted from previous model in porous media. And here, basically, we assume that inside the axon, there are 2 water pools. So the cytoplasmic water or axoplasms in the green area, and the bound water or surface water here in this red layer. The T2 times of these 2 pools are very different because of the water, because the water here is interacting with the cell's membranes, and this T2 is more shorter in the same way that the T2 of the myelin water is more shorter than the intraextracellular water because it's basically surface water. The bound water has a more shorter T2. And then these 2 pools are in fast exchange because for the echo times and diffusion time that we are using in the experiment, the water molecule have enough time to collide and to change from one pool to the other multiple times. So basically, this means that we cannot separate these 2 times, but we can measure it's just single T2, unless we use ultra short echo time, but that is not the case here. And the thing is that the apparent interactional T2 that we can measure here is the weighted sum of the T2s of these 2 different pools weighted by their volume fractions. The volume fraction of this pool is more higher, so this should be like the dominant contributor to the measure T2. But the thing is that the T2 in this pool is so short that somehow the contribution is not zero. So it's affecting also the observed T2. And the interesting thing to consider is that the volume fraction of this pool is related to the surface to volume ratio. And if we assume a cylindrical symmetry, then this depends on the radius, on the average radius of this axon. And this is the way in which we relate the interactional T2 time, the cytoplasmic T2 that we don't know, we will need to estimate it, and the inner axon radius. And then to estimate the histological radii, we conducted with Giorgio Innocenti, Professor Giorgio Innocenti, and Roberto Caminiti. Basically, they measured axons in many different rows in a postmortem brain in this 11 sector of the corpus callosum. And then we have 2 different histological samples. The first samples includes 4 rows, and we have thousands of axons per row. So we really have a good characterization of the distribution of radius in these rows. And in the second histological analysis, we have all the 11 rows, but the number of axons per row is smaller. So basically, we use the first sample to calibrate the model to estimate the cytoplasmic T2 and the surface relaxivity. And we use the second sample to validate the model, to see if we can predict the axon radius in this another ROIs. And then we also estimated not the mean radius per ROI, but the effective radius, because it's the radius that we can measure using this type of diffusion relaxation model that is in general quite different to the effective radius that we can estimate using diffusion MRI data, by the way, but I will not enter into this detail now because it's not relevant now. And these are some of the results. This map shows the interactional T2 times, the interactional T1 times, and this linear factor that is proportional to the interactional volume fraction. And the range is the range of values is within the expected range in general for the white matter. Then in this picture, I am showing a plot where in this axis we have the inverse of the effective radius, histological radius, versus the inverse of the interactional T2 times. As you can see, the linear correlation is nearly perfect. So basically, is one of these point here is the mean value per ROI in these 4 ROIs that correspond to the first histological sample. As you can see, this line is actually the surface-based model that we are employing. So basically, the intercept of this line is just this term. So from this term, we can determine the cytoplasmic T2, and the slope is just this term here, the surface relaxivity. So the surface-based relaxation model is predicting quite well the T2 values as a function of the histological radius. And we estimate these values. The T2 of the cytoplasmic water is 130 milliseconds, and the surface relaxivity is about 1.2 nanometer per milliseconds. And then we use that values now, because from this equation, so the thing is that if we know these 2 values, now if we know that the interactional T2 time in any other voxel, we can invert this equation to predict the effective radius. And this is basically what we did. Now, we can estimate the effective radius from the interactional T2 time in any other region using the parameters calibrated in the first histological sample. And now we did the prediction. The correlation is 0.68 more or less. It's not perfect, but anyway, we are not expecting a perfect agreement because these are 2 different brains. So we are just searching for associations. So we have an in vivo brain and an in vivo data. So these are different brains. So this the prediction cannot be perfect. But anyway, it's significant from the statistical point of view. It's significant, and importantly, the intercept is really small. So this means that we are not overestimating the axon radius, as happened with diffusion MRI in general. And then we repeated the same experiment, but now using the interactional T1 time instead of the interactional T2. And basically, we also got a nice correlation, but not as strong as the correlation we obtained using the interactional T2. We estimated from this linear equation the cytoplasmic T1 time and the surface relaxivity of the T1 time, that is 10 times smaller than the T2 surface relaxation. And then we also predicted the axon radius from the intra-axonal T1 times, and the results were more or less similar to those found for the intra-axonal T2 time. And then finally, we acquired more datasets, 3 subjects, and also we are also reporting a second histological sample where we have basically the mean values of axons from 9 subjects. In general, we can see, for example, that the histological radii from the 2 different samples do not agree, and this may be explained by considering that different histological procedures can create different shrinkage factors, and this can modify the values. Actually, these axon radius are like 25% smaller than the values found in the histological sample that we were analyzing first in this study. But anyway, what we can see is that both histological samples and the axon radius predicted from the interaction T2 times follow the expected pattern of low, high, low axons diameters that is reported in many anatomy books. And this is the estimation, the prediction of the axon radius in the entire white matter. We can say that we get like a symmetric pattern, and that is very similar across subjects. Not identical, but it's quite similar. And these values in general are much smaller than the radius predicted using pure diffusion MRI models. So this means that perhaps with this type of technique, we are estimating axon radius below the current resolution limits imposed by the gradient strength that we use in diffusion MRI. And then the main conclusion for this work is that the interactional relaxation times can be measured with the spherical mean MRI signal using a very high value, multiple echo times and inversion times. These interactional relaxation times are highly modulated by the histological axon radius in the corpus callosum, and that the simple surface relaxation model can explain the dependence and can be calibrated to predict axon radius in other regions. And this is a work that is now under review, and I would like to thank the whole team that participated in this study. It's a collaboration between different groups, including the people from Cardiff, Mohammed in Roche, of course, and the team here at the EPFL. And finally, I would like to show you quickly ongoing work where we are validating this technique in biomimetic phantoms. Basically, we have these axon mimicking microfiber phantoms that were generated using collectrospinning technique. And this is scanning electron microscope images showing the fiber morphology of this sample. It's not white matter, but it's quite similar in general. No? And basically, we can use the same type of acquisition to estimate the radius of this, the internal radius of these fibers. We have basically 5 different phantoms with different radius. And this plot shows the comparison of the effective radius estimated using scanning electron microscopy versus the radius predicted using this T2-based mapping technique. And you can see the line, the regression line is quite similar to the line of identity, showing like a perfect agreement between both estimates. And finally, I would like to thank you and also the Swiss Natural Science Foundation for the grant that is allowing me to work on these topics. Sorry, thank you.